Hi, welcome to Snakes and Adders. I know it might seem strange having a piece of card sat on the table, but we're actually going to look at just basic genetics today, see if we can help people understand quite what's going on with these animals and the morphs that appear on the market. There's usually a pair of alleles that will deal with eye colour, genetic inheritance of a disease, or in our case, pigments and patternation. So what we'll look at today is just the basics of putting these together, working out how we reproduce it. So a normal snake, for example, found in the bush would normally be represented as this. And say we were in the bush and we found a little albino that had just hatched out. Birds are going to eat it, or prey, so predators are going to get it, and because its camouflage is knackered, because it's got no melanin, for example, so it's bright yellow and white, so get picked off in no time. But we've been lucky enough to catch it before it gets eaten. And the way that that would be represented is this. A capital letter is the dominant gene. The small lowercase letter is the recessive gene. Say we were to take these two animals. For argument's sake, the albino we've just found is a male. So he's 2As, and the female that we've got is a wild type, and we, just for the sake of argument, know definitively that she's not carrying any genes. What we do is we work our way along and write down the combination of the two genes in this, which is called a Punnett square. So the results for this litter is that every baby is born a carrier. So a carrier we call a het or heterozygous. The dominant gene stops you from being able to see the recessive gene. Whilst ever there is a dominant gene there, we can't see it. So now we've got to look at a way of reproducing these albinos. Say, for example, we took a pair of hets. For a long time, a lot of morphs were very expensive, and this was the only route that people had into starting to try and produce their own morphs. So now we have a capital letter and a lowercase letter, a capital letter and a lowercase letter, representing that we have got two heterozygous animals that we're going to try and breed together. So the first 25% will be normal, they won't be carriers. The second 25% will be heterozygous. Third 25% will be heterozygous. And as if by magic, the last quarter will be the albinos. We've reproduced it. So the albinos we can remove. We know that they are what they are. The issue we have is marketing and selling the remainder of the litter. What we do is we take the remaining 75% as a whole, and we say that that now represents 100% of the litter. And of those, 66% are possibly het. So of the 100%, 66% are possibly het. This is something that you will see in shops all the time, 66% het. This is how they manage to work out that it's 66%. They're not just trying to pull the wool over your eyes or bullshit you. This is actually, the way that we work it out. Say, for example, now we take a visual and we put that back to a het. We're now going to get, because of the capital A, it's present, it carries, 50% of the litter are going to be hets. And they will be 100% het. The definite hets, they're carriers. The 50% only means how much of the litter actually was heterozygous. The other 50% are visual albinos. So this obviously creates more albinos and more animals in our litter of the mutated colour. So you could use this for recessive morphs in royal pythons, corn snakes, king snakes, Burmese pythons. So the list goes on. What we might end up with is where we've got two genes. So say, let's take for example an albino granite Burmese python. Albino is a colour difference, granite is a pattern difference. So that would be represented. For example, if we keep albino as A, well A, because it's recessive and it's showing. The granite part would be B. So that represents the second gene. Where things get slightly more complicated is we've got to work out how we reproduce this. So what we've got to do is work out our Punnett square. I'm conscious that the video might go on, so I'll try and rattle through this as quick as I can. This is how we work out the double genes. So you can see that we've drawn a Punnett square, it's bigger, and what it gives is each permutation of the two genes, whether it be completely normal and it's not carrying the gene for either, heterozygous for granite, heterozygous for albino, or carrying for both. So this one is AABB. This is AABB. This is AABB. And this is AABB. See how we're working this out? Hopefully it's making sense. So now we've got AABB. We've got AABB. There we go, you see, it's coming together. And the magic one. There's 16 squares. 1/16 of the litter will be the double representation of albino granite. What you've got is various other bits and bobs within here. So AABB, normal. AABB, that's het granite. See if we can find the opposite one there. AABB, het albino. Okay? So this is where it all starts getting a bit weird and a bit complicated. But you know, if you use a bit of common sense and we work it out, you know, you can sit and work anything out. Education's your friend with genetics, you know. Do some reading up and it'll help you along with the way that you understand. Where things get a bit weird with the genetics is we're going to deal with codominance. So this is quite prevalent in royal pythons. And for example, pastel is codominant, which means that now AA is still normal wild type because of the dominance of the pastel gene. The het carrier form now shows a colour difference. Pastel. And this is its full form, super pastel, even brighter, even more yellow. So this speeds up the process by which we can produce colour morphs, double colour morphs, triple colour morphs. because we don't necessarily need the supers to get a colour difference, which means that we can combine stuff. So take, for example, Bumblebee, which is a spider and a pastel. You breed a pastel, you breed a spider together, you can produce Bumblebee in the first generation. So, you know, the codominant side of things is interesting. And it was alluring in the beginning because obviously we're getting rapid results straight away. The issue is because of the rate of reproduction and replication of colours and the combination of colours, invariably the value that the animals attain in the first instance is fleeting and doesn't last that long. So I hope that that's been useful, and it might just give you a bit more of a clue when it comes to genetics, and there'll be more videos to come. Cheers.